Okay, good to see you all. So today the lecture will be about mutations and evolution, and it's going to be the last lecture that I'll be presenting. The next sessions are going to be with you presenting your ideas and experience your 5 minutes of glory with the whiteboard. I know it's a bit intimidating, so we give you another extra week to just absorb it, and we'll start not next week, but the week after. Okay? We sent you like the whole drill of how it's supposed to work. I hope you're finding somebody could serve as your mentor. If you have any questions, you have Sam and Lior happy and willing to help. We want you to be prepared, so make sure that you do it at least a week before. Send us what you've learned. If you find there's any problem or something like that, you can change your subject, you can change your date. We'll be facilitating. We just want to make sure that you're at your best possible in terms of preparation for when you're coming and doing it. We also opened up another date because some people are on reserve or other reasons. So we'll be having like a later date on the 25th of March. So it's like a month and a half from now. So there's going to be 4 sessions, coming up after not next week, the week after, and then a week of break, and then another special session. So if in case you find that you do need to change or need to think some more or something, we're open to that as well. Okay? In any case, make sure that you have all of those dates written for you because you're supposed to, even if you're not presenting, everybody should attend and then give feedback. We'll tell you the whole drill. Afterwards, it's also fun. Apropos that, you're also welcome to invite other people to come. Let's say people from your lab that you want to hear what you're going to do, your family, your friends, everything is good. And actually, they might, some people find it interesting, as I hope you will too. It's very dynamic and different stuff and things like that. We'll also be sending you today a short survey that is helpful for us in order to try and improve the course. For example, we're trying to make all those modules of things that I'm doing. Every lecture is built out of a few modules to make them publicly available as not on YouTube, not just as 1.5 hour lecture on something, but also like 10 minutes on some specific thing. So we'll be asking you which one do you feel that for you were useful, memorable, something you think is of special interest and things like that. So please help us with filling the survey. Nothing, 2 minutes and I think you're done. But you also have place to fill in some, general thoughts, suggestions, anything else. We'll be delighted if you do that. It's all anonymous completely. And those who would give us the best ideas would win a book. It's a book that I just got, which I'm really happy about. It's called The Biggest Footprint, 8 Billion Humans, One Clumsy Giant. It turns out it's a book geared to children, but it's actually intelligent children. And it turns out it's quite rising in many ways from the work that we've been doing here at Weizmann. So that's another nice thing to see. So if you want to see what you might be winning, if you have good ideas in the survey, I'm passing it by. And you can, if you want to grab it to your child, that's also fine if you have an intelligent enough child to read it.
But if it's anonymous, how will you know who to give it to?
What?
If it's anonymous, how will you know who to give it to?
That's a good point. I didn't think about that. If you think you're eligible because you gave a good idea, write your name. Or something. I know, if you have a better suggestion, let me know. That's a good point. Okay. Anything that I forgot? Yeah, we'll be recording. We'll be continuing with the recordings. But don't worry, we don't put it online to anybody. We just will send it to you if you find it useful. Usually it's a nice memorabilia from the course. And if there's somebody on reserve or something that can't come, we might share it with them. And if we find that we want to also put it as an example for next year on YouTube, we'll reach back to you and ask you if you're willing to give your permission that we do that. Okay, so that's our privacy policy. Good. Anything else? Let's get going. So I want to start with a sanity check. So what is a sanity check? Let's give you an example. Maybe that would make it clear. I thought it would be clear by this point, but I hope now it will be clear. I like to hike in Israel, other places. And I'm very surprised. I also like to have people, like a guide tour, like a geologist, explain to me what am I seeing. And I'm always kind of surprised by what they're telling me. Let's say I'm going to this beautiful place called Nahal Tzin, or somehow in the Judea desert or in Judea hills next to Jerusalem. And you're walking in this canyon and you see these huge cliffs. And, my geologist friends usually tell me that this is all made out of tiny microscopic organisms that, sedimented at some point, in some old sea that we had here. And all this is coming from them. And this does not make sense to me intuitively. How come from this you build this huge thing? And in order to do that, what I like to do is kind of like a sanity check to see if that makes sense or something is strange here. Okay, so that's an example of a sanity check. Let's try and do it together. Okay? So I'm walking, let's say, next to Nahal Tzin, next to Sde Boker, or somewhere next to Jerusalem. And what am I seeing there? How big is the cliff? Yeah, so something usually a few hundred meters. Okay? So let's just push it to the limit. Let's say it's a cliff of 1 kilometer. Okay, and there are such places that you might, maybe not in Israel, but you might even see a sedimentation or like something that you can see that's like a kilometer in height. There's also off the coast of England, the Dover cliffs, etc., and some beautiful places in Ireland, etc. Okay, so the next thing, what else do we need to know in order to do the sanity check?
The time.
Yeah, something about how long did it take to accumulate that. So what would they tell me? What do you think?
It depends. Yeah.
Yeah, they would say it depends, but usually they would give me all sorts of names. No, that's from the Permian, or that's from the, all sorts of Devonian. The thing is, it doesn't tell me anything. I don't know all these names. There are all sorts of parts in the world where they discovered those rocks, usually in England, and they decided that would be the name, and geologists know it by heart, but I do not. So I usually prefer to know it in millions of years. And usually it's some tens of millions of years ago, 30 million years ago, 70 million years ago, 20 million years ago, something along those lines. So let's say that, this 1 kilometer of, that I have here of, I'm walking here. I'm looking up at this. This is 1 kilometer. And it's been accumulating over, let's say, 100 million years, or so they say. Okay, usually a few hundreds of meters and a few tens of millions of years. So just to make it easy and simplified, let's do it that way. Okay, so if I have 1 kilometer in 10 to the 6, 10 to the 8, so 10 to the 8 years. What does that mean about the rate of accumulation? So the speed of accumulation, that's 1 kilometer every 10 to the 8 years. Does that make sense or not? So it would probably help me to make sense if I moved it into units that are more intuitive. I'm not very good at thinking of millions of years and kilometers. So I'll move from, I want to get rid of the 1 kilometer. I think the relevant thing here is microns, which are, because the thing that is sedimenting is like tiny, cells basically. And cells work in sizes of microns. So 1 kilometer, that's 1,000 meters. Every meter is a million microns. So I have here 10 to the 9 micrometers. What did I do here? Just using unit conversion, I have multiplied by 1. So no problem here. And we could leave the years. So this gives me about 10 to the 9 micrometers in 10 to the 8 years. So about 10 micrometer per year, or about 1 micrometer per month. Right? So that means, what's the size of those cells that are gonna, that are consisting this? Yeah, so anybody remembers names of these things that, are creating those cliffs? Somebody coming from, marine biology or something like that? So they have these names such as diatoms, foraminifera, coccolithophores, all sorts of things like that. Maybe you'll meet those names at some point. Usually they're a few microns in size and have these skeletons and all that. Most of them is made of water, like, 2/3 or whatever. So that means that, even if they're few microns, after they crush, and it's about 1 micron, something like that. So it means that in order to create that cliff over those geological timescales, you only needed to have one of the sedimenting every month. So for me, that passed the sanity check. Okay, that makes sense. I don't know exactly what's happening there, but if you told me it's every month, no problem. Every day might still work. If you told me there's it required a meter per day, I would say, no way. A millimeter per day also sounds a bit too fast. But a micron every month, no problem. So that's the essence of a sanity check. It doesn't tell you whether it's right in the end. I don't know. And probably it changes and the sedimentation rate and all sorts of things could happen here. But it means that we're roughly, it could happen. It could make sense, which is a sanity check. And you'll be surprised how many times it sounds very crude, and it is crude. The thing is that if it doesn't make sense, and even though you did it in a crude way, that tells you something is strange here. Usually either somebody has a mistake, or that you need to improve your understanding of things. I don't know what would be the case here, but it could be something that you learn from. Okay. Good. So this is kind of what we also, expect you to be able to do, either in exercises or in your life afterwards, and that's why we're spending time on it. You can also learn from this that geological timescale is kind of, it's not very intuitive to us, right? Because the thing that was striking to me, this huge thing, I couldn't see it happening. That's because I'm kind of stuck in the timescale of my life, of how I usually see things. Usually if they don't happen after a day, then I think they don't happen, right? Because we're so connected to, seeing it ourselves. If it doesn't happen over 100 years, then no way it's going to happen. But, the Earth has more time than us, and therefore that's why it's surprising, but that's why numbers help us understand things better on what's happening in the Earth system. Good. So also in that spirit, let's move towards thinking about evolution, which is also something happening both on the planetary timescale, but also on the lab timescale. So let's see what's happening there. So going to the lab and what happens there. How many mutations accumulate in an E. coli cell after or during an overnight of growth. So what do I mean? I'm taking a cell, and usually, we do it a lot. We put cells, for overnight because we need to, extract the DNA, do some manipulation, learn something new. We need to have a lot of cells, so we're putting them, and we're giving them time to replicate. And then I'm curious to know if I'm looking at the cell now versus what it was before, how many mutations did it accumulate over its genome? Question is clear? You want to do some short survey. Let's say, is it about 10 to the 3, 10 to the 2, 10 to the 1, 10 to the 0? Much less. Question is clear? Okay, who has the intuition of about 10 to the 3? 1, 10 to the 2. Okay, about 10, 10 to the 1, most people, 10 to the 0, one person, much less, myself. Okay, so in preparation for that, let's take, we'll build into that in kind of like a divide and conquer. And then let's ask a similar question. Looking now, like how would you approach this? What do you need to know in order to be able to solve this? Yeah, so we need to know something about the rate of mutations, yeah?
Maybe how big the genome is.
How big the genome is. Okay. How many replications in overnight. Good, so we need to talk about all of those things. So let's first just look at how many mutations happen in one replication. Okay, so just kind of in the divide and conquer, we'll get to the issue of overnight. For that, we need to know also the number of replications. But let's first of all look at like one replication of the genome. Okay, so for that, what do we need to know? We need to know, as was mentioned here, the length of the genome. And okay, good. So the mutation rate. What are the units of mutation rate? It's good because it could be a bit vague. So it's mutations per base or per base pair per replication. Okay, so I have my whole genome, I'm looking at a specific site, and I'm saying I'm now replicating it. How many mutations would happen there before the next generation of bacteria? Looking at that specific site after one replication. Okay, let's start to conquer this thing that we divided. We'll do this, and then we move to there. So anybody know something about the relevant genome length?
5 million.
Yeah, something like that. So bacteria, like E. coli, is about 5 times 10 to the 6 bases, 5 million letters. Okay, which means that if I need to replicate it, how many replications do I have? Is it 5 to the, is 5 million? Seems like I'm asking a tricky question, right?
Is it like one replication and then you have a double? Is that what you mean?
Okay, yeah. So this is roughly the number, but actually it's a factor of 2 extra because I need to separate the DNA and I need to do a replication on this side and on the other side of the helix. So I have about 10 to the 7, bases that I'll need to deal with altogether. Okay, what about mutation rate? That's base pairs, right? Thank you.
Fundamental something 1 to the 10 in the 9, something like that.
Something like that. Okay, so that's the kind of thing that it's very difficult to derive it from basic principles or to remember it from junior high school because you don't learn these things. But it's also a number that's actually useful to carry if you're going to do all sorts of these street fighting analysis of things. And so let me give you that number. It's about 10 to the minus 10 mutations per base per replication. Pretty low, right? Yeah, pretty low. And now it's actually similar in bacteria, in human cells, in yeast. It's kind of like, it's almost like, it's not constant, but it's a good value to carry in your head also, across the tree of life. And you can do all sorts of fun things, like for example, the kind of things you could do in the final part is to say, okay, if this was my error rate, if I wanted to write, I don't know, Encyclopedia Britannica, how many mistakes would I do?
That means 1 in 10 billion, right?
What?
1 in 10 billion bases get.
This is 1 in 10 billion, right? That means that like if we took all the people in the world and let each one of them write one letter, only one person would get something wrong. Or like we could do all sorts of fun things, but like let's do also fun things, but also applicable to our problem. Okay, so if we have this mutation rate, and this is the number of bases. By the way, to achieve this pretty low, as you say, like 10 to the minus 10, it requires evolution to work hard. Not only is it like just trying to fit the right complementary base to every base that was originally there, every nucleotide or whatever. It also does something called proofreading. Anybody knows what proofreading is? Yeah. Okay, it's kind of like, like when you're finishing writing something and then you give it to somebody else and tell them, read everything. If I had a mistake, please correct it. And people do it when you're writing books, writing, newspapers, whatever, because that makes you, and the cell does a similar thing. And they have a really cool way to do on the machine of doing the proofreading. It invests some energy, really interesting. And so originally it's not 10 to the minus 10, it's like 10 to the minus 6 or something like that, just from the combination. But then this extra mechanism of the same DNA polymerase machine brings you to 10 to the minus 10. It's interesting, by the way, if somebody has statistics on what's the rate of errors, let's say in newspapers or in books or something like that, I'll be curious to see. Because I'm not sure, like, is it higher than 10 to the minus 10, lower than 10 to the minus 10? Okay. At least in the thing that I'm getting for my students, it's higher than 10 to the minus 10, but that's fair. Okay. So given that, how many mutations happen in one replication? So now when we divide them to conquer a thing, now things are pretty simple. You just need to take the genome length, times this, and you get the answer, which is 10 to the minus 3 mutations per, what are the units now? Replication. Okay, base per and bases fell off. So what does that mean?
So 1 in 1,000 bacteria mutation.
Yeah, so if I take my bacteria after the replication and I look at that specific, at that bacteria, the genome is identical to a very high chance. Okay, let's look after an overnight of growth. After an overnight of growth, what am I missing from this? So I just need to get rid of this, of the replications, right? Because we're asking about, after all those, how many replications did they have? So yeah, we could do this with the 20 minutes. We've been to that at some point. So you could either do it that way. Okay, let's just do it. Let's say if we do it that way, 20 minutes, how many hours am I talking about? Let's say 10 hours. So I'll have 20 minutes mean 3 per hour. So I have 30 replications. Okay, and now we could dispute whether it's 30 or 25 or 35 or 20. Okay, I don't care. Because if I have 1 in 1,000 chance of a mutation per replication, it means that even after I'll be multiplying by the number of replications, which is 30 or something like that, I'll still have something that is much less than 1. Okay, so I was the only one who was right. Good for me. Okay, fantastic. So the issue raised here is about the number of bacteria. So let's see if I wrote the question correctly. Okay? How many mutations accumulate in an E. coli cell during an overnight of growth? It seems to me like you're interested in a different question. Right? So if we solve that, that's good. We could also ask the question, how many mutations accumulate in a culture, in the whole culture during overnight of growth, and then we should take that into account. Okay, so it's a different question, which is actually not a question I'm not interested in. So let's say the following. Okay, or let's ask it. Okay, let's do the following. I need a specific mutation at some point in an equalized cell. Specific, okay, so for example, I want to have an A turned into a G at a specific base. Okay, and why do I need this? Because this is going to save my PhD. You know, if I have that mutation, I could test whether this gives antibiotic resistance or new metabolic capabilities or changes something that I'm curious about. And this is, or let's say, if I have even one of those, you know, if I create that, you know, that would enable them to grow, and so far they didn't grow or something like that. And now the question is, what do I need to do? Or like, okay, and my options would be, like, what are ways to make mutations or to make them happen faster or something like that? Anybody knows any methods for that? What? Okay, so one option is to say, let's use error-prone PCR. Error-prone PCR on my overnight culture. This is a symbol for overnight. What else?
Design plasmids with a mutation inserted.
Okay, so insert it using, let's do it with CRISPR. It seems to be very popular these days. Any other method? Okay, so there's like, you could use X-ray or UV. Okay. Okay, we could also do mutagenesis. Okay, let me give a fifth option. Do nothing. Why do I suggest also this option?
Because there's a natural rate of mutation occurring.
So maybe just based on what I already have in the culture, maybe I'll already have it. So let's check on this, okay? So what do I need to do in order to calculate that? On the one hand, I need to know my rate of mutations, which we just calculated. But this is per...
about the mutations in overnight. So you have 1 in 1,000 bacteria that are mutated, but then you have overnight you have 2 to the power 30 bacteria that are made. So it's really less than 1.
Yeah, so that's a good point. That's exactly what we're gonna. So that's kind of like goes also with what Yarden was asking before, and what we'll be analyzing just now. Okay, so let me do that, and then we see how it fits together. Okay, so we had 10 to the minus 3 mutations per replication. Okay, and we also know that we have about, let's say, 30 replications in the culture overnight. And now we need to also plug in the number of cells, as was pointed here and pointed by you. So how many cells do we have? Yeah, so I think we calculated it at some point in terms of the OD, and I think it's about 10 to the 9 per ml per unit of OD.
I don't get why you choose to represent it as 30, because every subsequent replication, there are more replications that take place. The first cycle there were 2 bacteria being made, and the second cycle there were 4.
Yeah. Okay, so
The more you progress, the more you get a chance for mutation.
okay, so let's do it the following way. Okay, there's actually different. So one thing is to look at, like, let's say we started with one cell, and we had one replication, and then there's 2 cells and another replication, and 4 cells, etc. And then when we reach 30, we'll have about 2 to the 30 cells. And we could do the summation. And the depth of this would be 30, and you can calculate that way. It's true that there's another way and the trick to do it. It's just less intuitive, but I'll go with it just because you raised it. And actually, it's also pretty elegant. If you ask how many replications would happen in general here, starting from, let's say, 1 cell, and finishing with how many? Let's say it's 2 to the 30 cells. How much is that? So 2 to the 10 is 1,000. Okay, so it's like 1,000 to the third, so that's 10 to the 9. 10 to the 3 to the 3, it's about 10 to the 9 cells.
Can we think of it more in terms of the amount of food available overnight? Because they can undergo, but then they are limited by the food they have. And most of the time they run out of food by the overnight culture.
Yeah, so you can try and also do it that way, and you'll get a similar result. I just know that this is roughly also true for being the number of cells per ml per unit of OD. So, from different angles that we could do it, I could tell you that's roughly the right order of magnitude per ml. And usually OD, and like an overnight would be, a few OD, a few mLs. So usually you would have about, altogether, about 10 to the 10 cells. And just 1 second, guys, let me finish so everybody could be on top of it. So we have about 10 to the 10 cells that I'm working with. Okay? And this would also be the number of replications. Okay, which is a tricky thing. It doesn't matter if they replicate in the beginning or replicate in the cell that I'm picking, whether it was born now or in the past. Overall, there's been 10 to the 10 such replications in the culture. Okay, if it's a bit counterintuitive, sleep on it. Okay. No, just 1 second. We have 10 to the 10 cells. So now we could just do it pretty elegantly by saying that because we have this number of replications, and this is the number of mutations per replication, it means that overall, I have about 10 to the 7 mutations in my culture. So just 1 second, please allow me a second. So we have about 10 to the 7 mutations accumulating in the culture altogether. Okay? Which if you remember, this is also roughly the number of bases or base pairs that we have in the genome. So it means that, unless, something is strange, choosing to have all the time, just mutation to a specific site, I'm sort of like covering the whole genome with mutations. So roughly I have all the mutations possible in the world in this genome, all the single base pair substitutions, just already there in my test tube.
But it's not in one cell, it's across all cells.
What?
It's across all cells.
Across all cells, exactly. So now it's not in a specific cell, now it's in the whole culture. So you might feel maybe I tricked you, but I didn't trick you. It's just like complementary views. In every cell that I'm taking after an overnight, if I was able to sequence just that specific cell, most chances are there's no mutations at all. It's the same as the original. And at the same time, if I look over the whole population, because there's just so many cells, in each one there's only 1% chance that I'll see some mutation. But because there's so many cells, I'm actually all together with all those 1% somewhere, I'll be covering kind of exhaustively the whole genome and have all the mutations possible. So both things are true. Yep.
Doesn't that mean that you only have one mutation per cell? Like, can't we have multiple mutations in a cell? And then it wouldn't necessarily cover the whole.
Yeah. So if I understand correctly, the question is about the fact that, maybe there's 2 mutations happening in the same cell. Okay, so we'll even talk about that in a few minutes. That's also fine. That could also happen. But the chances for that are lower. So now it's like a geometrical, whatever.
So when you said less than one, you meant looking at a single specific cell at the last generation?
Not when I was talking before, when we said like overall,
Yeah, less than one.
Yeah, so doesn't matter if it's just, if I'm looking at a specific cell, either in the last replication, at that specific location, or even just over the whole genome, almost nothing, 10 to the minus 3. If I'm looking over all of its replications overnight, also significantly less than 1. Let's say 1 in 30 or something mutation. But now let's say 1% of getting a mutation. If I repeat that 1 billion times or 10 billion times, in the end I'll get everything. Okay, so it seems like there's a lot of food for thought here. So maybe it's a good time to take a recess and try in your head to run these two complementary perspectives. So we continue our exploration of mutations and evolution. And I want to move from those tiny bacteria cells that we played around with so far to think about humans and what happens with our mutations happening to humanity. So let's think of a very similar situation, but now I need a specific mutation at some point in a person. Meaning that I'm interested in a single base mutation, and now the genome that's not long enough. It could be even like this, because it's really a much larger genome. And now the question is, kind of like how we looked at before, how rare is it? Right? Like if I'm looking for such a mutation, let's say it's a mutation essential in order to survive, I don't know, a new menace to humanity, some Ebola that, kills everybody unless they have that specific mutations. How difficult is it to find this? Can you try and calculate it now, given what we know already? Anybody is willing to try and do the calculation? So let's first divide and conquer it, okay? What do we need to know in order to be able to know if we have this mutation, let's say we need it to arise, in the most recent generation of humans. Meaning like it's like a de novo mutation. So for that I need to know, on the one hand, the mutation rate. So I have a mutation rate. And as I was saying, it's not very different between humans and bacteria. And we had here the genome length, which maybe I want to update for humans. How much is it for humans, the genome length?
9.
Right, so it's a few billion base pairs. So about, say, 3 10 to the 9 base pairs, or because of the factor of 2 with the bases, etc., let's say it's 10 to the 10 bases. So that means that I have about now 1 mutation per replication. Okay, so you take this whole long genome that each one of us has, and in replicating it, because it's so accurate, there's only 1 mistake over those 10 billion letters that are being put there. But that's not what I'm looking for because of 2 reasons for this problem. Why? There's 2 differences here. One is that this is, that's a mutation somewhere along the genome, but I was curious about a specific mutation. Okay, so I'm looking at just specific mutation. And there's another thing, that is that this was calculating it over replication. And let's say if I'm looking for a person, there's more than one replication happening between one person and the next, and my daughters. So that's also something I'm interested in trying to correct.
You also egg cells rather than somatic cells.
Right. So in a way, you're interested between those gametes, or like the thing that your sex cells, and the next sex cells. In terms of how it's getting propagated. And the question is, how many replications happen along those lines? Let's talk about it in a minute. Okay. Good. So let's start with the first thing. So we have one mutation per replication, but we said I'm interested in this specific location. So in this specific location, it doesn't help me to multiply by the number of bases. I'm really interested in the mutation per base per replication. It's only 10 to the minus 10. So it's a very tiny chance to get that, except for the fact I need to multiply by the number of people around the globe. If I want to get some representative, how many people do we have? So we have about 10 to the 10, which is pretty nice. In a way, in a funny way, it exactly offsets this. That means that in one replication, across all of humanity, in every specific location, you're having a mutation, which is nice. It means that as a humanity, we could adapt to this Ebola. Somebody would help us do that. And that's just in one replication. But as was pointed out here, there might be more than one replication between my sex cells and whatever is the next generation. So let's talk about that for a minute. So how many replications do I get across a generation in a human? Anybody have a suggestion how to approach that?
We should find the replication rate of the cells inside the human and take the average lifespan.
Okay, so one suggestion is to say, okay, if I have a cell, let's see how long does it take until the cell cycle, which has one generation, and then I could see how many happens over a lifetime or something like that. That's kind of like the idea.
You don't reproduce over a lifetime in a specific period of time, between like 20 and 40, let's say.
Also,
It's not possible.
for women, like the egg cells are already there. They are not like newly made. They were made in the grandmother.
First, in males, they have the stem cells that are making the sperm, so they always renew themselves.
Okay, so this is, as you can tell, it's so like it becomes a bit messy. There's many different things happen here. So on the one hand, you could do it this way, but that's relevant for some somatic cells that usually turn over. But we're looking at those that either it be the oocytes for females or the sperm for males. So it's kind of like different in terms of the formation. We could, and you could think about, say, in two general models, I would say. One is kind of like this, and this divides, usually a normal cell divides about 1 per day. And then over a year, you'll have like a few hundreds. Over a lifetime, you have, maybe 1,000 or something like that. That's like one way to think about it. It's a few thousands over a lifetime. Another is to think differently and to say, okay, let's say I'm starting with one cell and I have like this division and this division and this division and this division, the whole tree of binary divisions. Then you can ask, okay, how many would I have what would be the depth of the tree? And then, we calculate for bacteria, it was, say 30 replications. Humans are bigger, but let's say you've learned about Ron Sender gave a lecture about how many cells there are. Instead of 10 to the 10, it would be, say, 10 to the 12, 10 to the 13. So not a huge difference. Another factor of 10 to the 3, that's another 10 replications or something. That would give you few tens to maybe 100. This would give you, I don't know, few thousands. So it's actually not, it's different. And actually, if you look at, what happens usually, it's some sort of a mix between that. We're not a binary tree in the way because some of the cells continue to divide, some of them stop to divide. Those that continue to divide look a bit more like this. But it's not exactly like this because it has this binary structure as well. So it's somewhere in between those. Okay, and then you probably wouldn't be surprised if I give you a value that's somewhere between here and there. Indeed, if you look at now, I'm giving you like the empirical evidence. It turns out it's different for males and females, as you guessed. And so for females, you make just the oocytes, and the oocytes are kind of like, it looks more in like this binary tree formation, and it's somewhere between 20 to 30 replications between, if you look between, you can think about it between the oocyte of the mother and the oocytes of the daughter. Okay, between my wife and my daughter, that would be roughly the number. On the one hand, on the other hand, you can look what happens in terms of males for the sperm. And sperm, there's way more sperm than oocytes. We make, millions of them, and we waste them, and then more millions, and that. So, much, much bigger numbers. And if you look at what happens there, it's usually somewhere between, if you look now between the sperm of one person and, the next generation, it's somewhere between 40 and few hundreds replications. What do we learn from this? It means that if somebody has a genetic problem, genetic mutation, whom should they blame? Yeah, so they could blame their father at a much higher chance than their mother, which, useful. At the same time, by the way, it's actually a bit more complicated because this talks about a number of replications, which lead to, all sorts of things, like what we analyze here, like base pair mutations. It turns out that, one of the mechanisms is recombinations, which is a different mechanism, and that's actually higher in females than in males because of all sorts of reasons I don't know. So in general, it's just more complicated than this. But you see that, like, in general, what we're talking about, it's a mechanism somewhere between here and there. And, we could have spent, days on days, if not more, disputing whether it's this or that. In the end, I don't care so much. It's like, because if it's between this and that, a few hundreds or 100, and that's roughly also what we see empirically, it boils down, maybe for some other reasons, it's interesting to know exactly the mechanism. For a back-of-the-envelope calculation, it doesn't matter so much. We could say 100 replications per generation. Okay, so now armed with this, you see that to get this specific mutation, we have it somewhere in humanity because there's so many of us, even after one replication. That's what we calculated before. And between generations, I'll even have the extra factor of 100 or so from the fact that I will even have more mutations. Good? Great. So now I have the following challenge. It turns out that it's not enough to have this specific mutation to save us from this, Ebola or whatever it is. But it's not very far either. The only difference is that now I want to ask the following question. Instead of a specific, I need 2 specific mutations at some point in the person. So I need to have an A to G here, and I have to have a T to C at another location. Okay, so I have to have this and this. Okay, now I'm asking how rare is it? We have it in our country. Or someone in the world has it in the last generation, or nobody in the history of humanity had it. Okay, the options are clear. Let's say I need these 2 mutations to occur, and now I'm asking, okay, somebody in the country, have it, some baby born, in the past few years, or maybe not here, but somewhere around the globe, you'll have what's needed to save us from this menace to humanity, or that nothing's going to save us. Let's check on our intuition after we did some of those things already. So who's voting for A? Who's voting for B? 10 people. Who's voting for C? Another 10 people. Many people don't want to engage, or don't want to put their name next to it. I understand. Like after getting burnt so many times, you don't want to. But I think you're enjoying the fact that you're getting burnt. And okay.
This is not considering how we are exposed to all the mutants out there, and the rate of mutation could be much, much higher.
How does the rate of mutation could be much higher?
We are exposed to UV a lot. We are exposed to a lot of other chemicals in our everyday life, which is promoting mutagenesis. So we are not considering that. We are just considering.
So I think this. I think this rate of mutation is also what we observe in humanity also today. It might be, of course, if somebody, I don't know if you saw this amazing picture before they learned about radioactivity. They used to use radioactive material in watches in order to have them shine in the light. It's quite crazy. Like they would put them next to phosphorus or something like that. And you can see, like in the 19th century, you would have people sitting. It was mostly ladies that were working at the time, and they would put this radioactive material in the different 12, things so that you could wake up at night and look at your watch and it would be shining before you had batteries or something that could do that. And only a few years later, they understood it's actually dangerous for you to have such a thing anywhere. Clearly not next to you. Clearly not somebody working and doing that. So there could be all sorts of things increasing those rates, and maybe some pollution from the environment and things like that, exposure to you. I think this is like the rate I'm showing is also with normal exposure to UV from sunlight, unless you're sitting at the beach all day long and trying to get a tan.
About the reproductive cells, right? So does UV affect them? Protected,
Okay, so let's try and do it together from what we've learned. So remember, we had this for one replication, and we already updated for humanity. So we have this number of bases, this rate, which gave us this mutation per replication. And now we added in that I'm going to multiply it by about 10 to the 2 replications per generation. Okay? Which is, the things I just deleted there, but you remember. So from this, I can infer that I have about 10 to the 2 mutations per generation. But now we want to do it. There's 2 other things here. Like we're looking, remember, this was over the whole genome. And I want to get a specific place. Yeah, so in a way, I don't need to go through this times this, etc. I'm really mostly interested in this. So forget what I just written here. I actually want to look at the mutations in a specific base per replication. And I want to multiply it by just what I did here. 10 to the 2 replications per generation, which means that I would have 10 to the minus 8 mutations. Now I'm done with replication. It's per generation, per base. Per base, you can, it's like per site, like a specific location I'm interested in. Good. So I have this rate of mutations per generation per base. And now the thing is, I need not only 1 mutation, I need 2 mutations. So what happens if I need 2 mutations?
Square.
So I need to square it to get 2 mutations. So I have 10 to the minus 16 as my rate per generation.
Do you want those 2 mutations to be in the combination? Like there are so many combinations of 2 possible.
Yeah, but so yes, I need those two mutations occurring in the same person. Like I can't save the humanity if like one person will have the one mutation and another person have the. I need to have the same the two mutations in a single person. So
Also, you want
just one second, just one second. So I have 10 to the minus 16 chance per generation to have those two mutations. But now I need to multiply by the number of people that I have available to me. So I multiply by 10 to the 10 people in humanity. And I'm getting about 10 to the minus 6 per generation. So it means that I need roughly 1 million generations. So we're in trouble. I would need 1 million generations of all of humanity to get those two random mutations to occur in some person in order to save us from this menace. And you know, like 1 million generations, that's like 30 million years or whatever. That's more than the existence of humanity since we separated from the apes and all sorts of other friends of ours. What do you think?
I think nobody in history.
What?
So nobody in the recent history.
Yeah, so indeed nobody in recent history had it, you know, occurring randomly. But that's yeah.
We have immune system, and there is something different that's going on there with the mutation.
Okay, so that's a good point. We have this adaptive immune system that is able to deal with things in a different manner. That's a very good point. That could definitely help with the numbers.
I mean, we know we are here, and we are probably able to survive viruses.
Okay, so there is this thing that so there is also, it's good also to remember the facts outside, just, doing this calculation. Like we do exist, and we know that like there's been more than 2 mutations between us and the chimp. At least some of them, we think of them as adaptive or useful. So what's really happening here?
You're looking at two specific points, right? And you could have a wide variety of mutations, but some of them would be adaptive in a certain situation.
Okay, so one way to think about it is like, let's say we do want to deal with some specific virus or bacteria. It might turn out that the target needed for the mutations, there might not need to be a specific basis. Maybe the target size is different. Maybe somewhere in the genome you need to change. They have 1,000 different options, and another 1,000 different options. Then the combination is like there's 1 million combinations, and then you can do it much faster. Okay, so there could be such a combinatorics, but it still doesn't take effect from this. If you do need the specific ones, you're still stuck with this.
Also, there are places in the genome that are more prone to mutations than others.
Okay, so there are places that are more prone. On the other hand, there's things that are less prone. So it depends. If you need it in the specific places, on average they'll be as prone as any other. But it could be something along those lines. Yeah.
Also, some places in the genome can be bigger.
Can be.
Like some mutation, if you have it, you won't have any acceleration. So it's not so random.
Okay, so maybe it's not random in that sense. Okay, so that goes into the direction that I think is also very important, and that is that if you want to have something with 2 mutations, and it's worse even if you want 3 mutations, something like that are random, it's very hard to achieve them in terms of the combinatorics. The chances of getting them is like super low. What could help you?
Something that is not random, but directed.
Yeah, so if you have a directed way to do exactly this is great. But usually evolution doesn't work in that directed manner. But there's a different trick that could help you, which is kind of like effectively achieving something similar. What makes evolution still work?
Pressure.
Yeah.
Inheritance.
So it's
So it doesn't have to be de novo at the same person. they can have one mutation and then the other?
Okay, so even if you have just one mutation and it would be beneficial, then as we calculated before, beneficial, like single mutation, it's kind of easy. We get them in every E. coli, 5 ml that grew overnight. If there's a mutation that's needed and already has fitness advantage, it could, you'll have it, and from there it would overtake the population. Evolution has something to work with, if you like, even after one overnight. So if every mutation by itself would already have an advantage, then there's no problem having one in one overnight, and then afterwards another one in another overnight, and they would be accumulating. It's just this, the problem is if you want them to be independently, like completely random without having a selection for them. If you have selection, then the numbers could easily add up. Okay, so it's kind of a bit also counterintuitive, which is like what's happening to us this lecture. One mutation, not so difficult. Usually you could easily achieve it in the population. Two mutations, randomly, effectively impossible. Okay, we calculate for humans. For bacteria, it's not very different. Effectively impossible. But if you want those two mutations and you build them one on top of the other, like each one having some fitness advantage, then you can do that. There could be other things. Evolution is more complicated than what I'm able to present here. We don't even necessarily understand everything. But there's also the ability to get things through, transposable elements, recombinations, other mechanisms that could give you more than one mutation at a time, as long as that is in the target with what you want. Like it's not easy for them to achieve exactly those two, but if the thing that would save you against that menace is really some combination that you can do many things in the same time with a specific transposon fitting somewhere on some, duplication or deletion event, then you can do many such things happening at the same time. More thoughts or comments about that? Yeah.
And also bacteria can give each other beneficial mutations that they acquire. They can transfer genes and
Through conjugation
yeah.
or things like that.
Like I'm thinking to myself, okay, so if two mutations is almost impossible, then we can solve the antibiotic issue by just treating with two drugs, because the likelihood that they could be resistant to two at once is almost impossible, but it's not true because they can easily give each other, if one got it and a different one got a different one, they can give it to each other.
Okay, so that's a good point. That bacteria could also do, so it relates also the fact that that evolution often, why does it use sex as a way to make as something that's advantageous and makes us so happy about it? So it turns out it has this ability to recombine mutations that happen in different locations, and then you can, one mutation happening here in the mother, one mutation happening, in the progeny of the father, and now they meet together. So that could also have effects here, and bacteria can do something similar through conjugation. And that relates, for example, to think what happens if you have an antibiotic and maybe you're putting two antibiotics together. So about that, I'll just mention, so this thing of putting cocktails of antibiotics, that's really what helps us really deal with antibiotic resistance. So that is really a very powerful tool because the bacteria need those two things happening together, and that's very rare. And then they're fighting against us with mechanisms not unlike what you said. But it's also the case that the ability they are able to resist that is because also these things are usually not completely independent. What needs to happen, in order to fight one antibiotic and another antibiotic, usually if you have an efflux pump, something throwing it away, it works not badly against one and helps with the other, and all sorts of things like that. If it was completely independent and you do the cocktail perfectly, that would be good. The problems, for example, is usually when you say you want to deal with that, and you want to give the cocktail of those two antibiotics, so that the bacteria, but sometimes you're a bit sloppy. Meaning that in some cases, the person, instead of taking both antibiotics, it only took one. Okay, somebody took only one, or you gave it to pigs or to chicken or something, so they won't have antibiotic, and you didn't give the cocktail. So the bacteria easily adapt to one of them. And when they adapted to this, to adapt to the other one, that's again easy. So you have to be pretty careful about always giving them together. And we humans, we're not that careful in doing these things. So that's why these things, in principle, they could be a great solution. And in practice, often we're losing to evolution. But we're also gaining a lot from it. So I think it's all fair. Okay, so as you can tell, there's really in my perspective, interesting perspectives that come from this marriage of evolution and numbers. And you can also tell that I didn't tell you the whole story, because I don't know the whole story. Because clearly we are here different from the chimp. A lot of numbers happened along the way, and a lot of processes, etc. But I just wanted to give you a taste of this and how you can get more insight into such complicated phenomena with relatively simple type of analysis. And that's the spirit of the course and what you're expected to do next. Good. So, in terms of this lecture, I think we achieved what I wanted. So I think it's time to, first of all, congratulate all of you for making it through the whole set of lectures and preparing for you to present. I also want to take the opportunity to say special thanks to Lior and Sam, that are the teaching fellows of the course. Much of the things that you saw here were also, developed in combination with my good friend and collaborator Rob Phillips at Caltech. If anybody is ever going to the West Coast, he's teaching, similar things, much more elegantly and charismatically than I am in his course at Caltech. But you can see some of his things as well. And many of the things that were also developed together with teaching fellows in previous years, just like what Lior and Sam are doing. So originally, it was Maya Shamir who did it in the first year that I gave the course, and she was the brave person to give the first presentation in front of the class. And now she's finishing her postdoc very successfully. So somebody would have to be the first, but don't worry, we'll give you all the support, and it's not the end of the world. And afterwards, we had Yinon Bar-On, who's now finishing his postdoc at Caltech, and Ron Sender, that you've heard in one of the lectures. So I thank also them for everything here. And just before we finish, we had an idea that I don't know if it's a good idea or not, to do kind of like an improvisation of back of the envelope estimates. And the idea is that if you have something that you want me to estimate, we could see how well I can do it. I don't know if it would work. It's like the first time we tried that. It's kind of like the Weitzman version for reality TV in lectures. So I give you a minute to think about ideas, and then we'll see how that works. And we have another 10 or 15 minutes to try that out. Sounds good? Okay, so think about for a second what you want what challenge do you want to give me? Okay, we're open for challenges. There's usually okay. Maybe you want to say it, or?
I just wondered what weighs more, the stacking our fat cells or the membrane of all the cells in the body.
Ah, that's nice. Okay, so let's collect a few of these and see what so what weighs more? Our fat reserves or the lipids in our membranes. Did I get that right? Wow, that's an awesome question. Okay, let's see if there's more, and then we can jump into them. Anyone else?
How many traffic lights there are in Tel Aviv?
Okay. How many traffic lights in Tel Aviv? Okay, let's try and see what we could do about it. Which one you think is easier? I think Tel Aviv is kind of like easier to approach, but let's try to deal with the tougher challenge for now. So what do I know? So in terms of our body, in terms of fat, just from doing some your medical checkups every once in a while, or it's something about, I would say, somewhere between 10 and 20% for I don't know, for me, for somebody else. So I would say fat reserves, that would be about 10 kilograms per person. By the way, there's an interesting thing here. If you're thinking about the person that gains weight, is it that now they have more cells or bigger cells? And you could ask the same question about, say, like a plant that grows. Is it because the cells are becoming bigger, or and so if anybody wants to read, in my book, there's a chapter that talks about, this sort of thing. It depends on the person, or not the person, on the biological mechanism. For people becoming more obese, it's usually that the cells remain in terms of number, but just increase their size, kind of like what was just said here. But for growth in plants, it could be different. Okay, back to this. Okay, lipid in our membranes. So, what do I know here? So I remember that, say, for bacteria, E. coli, lipid is about 10% of cell dry weight. Now, humans are different. A human cell is different than the bacterial cell. How is it different? On the one hand, it has many more membranes, right, around in the ER, in the mitochondria and all that. And that leads you to more lipid content. On the other hand, it's much bigger, which means that the ratio of surface area to volume is actually such that, let's say, even at least the outer membrane would occupy much less of the weight or something of the cell, and therefore it would push to the other direction. You see what I mean? If I had to take, an overall estimate, thinking, what's happening to the ratio, not in the bacteria, but in the mammalian cell, I would probably say the same thing. About 10% of the dry weight. And why am I saying that? Well, it can't change by that much. In the sense, it couldn't be more than 100%. It's probably nowhere next to 100%. We know that we're not just lipids. And theoretically, it could go much, much lower. Like if I was just looking just at the cell membrane, because of what I told you about surface area to volume, it probably would lead towards 1%. But I think, this, because we have the mitochondria and all the ER, etc., I would say as order of magnitude, probably I'll stay with 10% of dry weight. Okay, 10% of dry weight, because we're, say, 70 kilograms, so the dry weight is about 20 kilograms, 2/3 water, so 20 kilograms, and then 10% out of that, it would lead me to say about 2 kilograms per person, which I think is good enough to say that this weighs more. Like, I don't have here a factor of 1,000, which would have made it sure for, 100, which would have been absolutely sure. 10, I would have been mostly sure. I have a factor of 5. I think I'm pretty sure, because I think that the uncertainty here is very small. That's like less than 2-fold. And the uncertainty here, it could be more than 2-fold, but probably not by a lot. And also I need to check chances. So I would say I think this is higher. This weighs more. Anybody knows the answer? Okay, so maybe it's up to you just to check, if I'm right. Okay, but I really like it. It's a nice way also to connect between like the question about microscopic things or like cell physiology, structure and cell biology, and something that we look from a larger perspective. And I like these things that combine these two. And let me know if I'm wrong, especially. Okay, we have time for the traffic lights in Tel Aviv. Okay, how do we do the traffic lights in Tel Aviv? So Tel Aviv has this nice thing that like it's actually one of those cities, well, it's not Manhattan, but it does have this, you could think about it as something, I could just think about it something like a grid. If I need to think about the grid, it depends on how exactly you define Tel Aviv, but let's say I would say it's, 10 kilometers by a few kilometers. Okay? Now, it doesn't have traffic lights on all the streets. Yeah, actually, okay. So let's do the following. Let's start with thinking that in each grid meeting, there's actually more than one traffic light, right? You usually have to have like an intersection usually has, it depends how you define it. Is it only for the cars, also for the people walking? Okay, so it has about, let's say, 10 traffic lights per junction. And now we need to see how many grid points do we have, how many traffic lights, and then we also have like a probability, like what percent of junctions has traffic lights, right? Because many times it's like this small street with another small street, there's nothing there. Okay? Good. So let's do this. So what is the effective grid of streets in Tel Aviv? So I would guess it's something like, 5 buildings or 10 buildings by 10 buildings, something like that. So let's do a zoom in on one of this grid. I'm saying there's like 5 to 10 buildings on each side. Each building is, 5 to 10 meters, let's say 10 meters. So this is about 50 to 100 meters each side. So this 10 kilometers gives me about between 100 to 200 streets. And this few gives me somewhere between few tens to 100 streets or something like that. It gives me about few tens of streets. Okay, so now it's a good point. I calculated it from basic things, but now I also know that I've walked in Tel Aviv for a while, right? And if you tell me that if I want to go to the, I get off at one of the train stations, which is on the eastern side, I want to walk towards to play volleyball on the beach, and you tell me I'm passing a few tens of streets along the way, I would say, yeah, a few tens of junctions, that makes sense. And if you tell me I'm starting from somewhere, I finish eating something nice in Jaffa, and I want to go to the port up north, and I have to go through 100 small streets, that's also reasonable. That's why I would take a bike. I would not walk it, I would take the bike. Okay, so altogether we have, let's make it just nice, 100 times few tens. So we have a few thousands of junctions. And we had about 10 traffic lights per junction. And then we have this fudge factor that I'm not sure exactly how to approach, of what percent of the junctions have traffic lights. I would tend to think somewhere between, let's say, 10%, somewhere between 1 and 10% would be my guess. So what do we get altogether? This gives us few tens of thousands, but then 1% of that would be few hundreds to few thousands. So somewhere between few hundreds and few thousands traffic lights. Somebody has a friend that works in the Tel Aviv municipality?
No, it's the junctions. I had
No,
for
no,
meetings.
it's
I read 5,000.
Yes.
Okay, so somebody, okay, so good, stay with me for a second. So somebody checked in Google, or in, what is it? It's a Ma'ariv, even better. Ma'ariv, the very high impact factor. So they said 450 traffic lights. Okay, interesting.
But I'm reading a committee transcript. ועדת התחבורה ובעיריית תל אביב.
Okay, we have the committee now. Tel Aviv.
2017, and they claim that they have 5,000 traffic lights and 431 junctions.
Again, what are the numbers again?
431 junctions, but overall number of traffic lights is 5,000. So it's about factor of 10.
So it's 10 lights per junction.
Something. Because not all the traffic lights are junctions, maybe, I don't know.
Actually, it works out well because it's about.
Okay, it's
10
just
lines
getting
per
better.
junction.
Okay, so a few comments on this. One is that, like, you could try and look it online, but actually without having, like, something to guide you, you can easily get a factor of 10. And also it's not, and you don't know exactly whom to believe in a factor of 10. Okay, so in general, I would say, both the 500 and the 5,000, they're sitting pretty okay, in my perspective, within this estimated range. And now I could also know how did I get to it, which I think there's something obviously more fun into it. And if I had to guess what's right, I would say that that's probably right. And here it might be that they were misthinking about this number. They were like this. They were thinking about junctions, but they wrote it as traffic lights, but actually meant junctions where you have traffic lights. Okay, and the fact that I got the upper limit was few thousands, and this is 5,000. I don't make any big deal out of it, right? Like it's easily I could get a factor of 2 for many different things here, and I wouldn't know. But if this thing I could, this also sits within what I was giving, but I could pretty easily say from all sorts of intuitive reasonings, and it probably is too low. Okay, you could also send me the grade of how well I did, but I think it was a good experiment. I think we might do it again. Okay, I think we're done for today. Thanks everybody. Looking forward to hearing you. No lecture next week. We do meet the week after. And no exercise. And if you still need more time in order to finish the current exercise, no problem. You can do it in the next few days. Let's say maximal until the end of the week, unless you need more time.